So today we are going to discuss the respiratory system. All right. So the idea is we're going to break down the respiratory system into both the upper and the lower portions of the respiratory system. We'll talk about its configuration and the anatomy of the respiratory system. And then we'll spend kind of the second half of the lecture discussing the physiology of the respiratory system, how it works. Okay. All right. So let's get started here. So the respiratory system, right? It's necessary because our cells need energy, right? So all of our cells in our body have this thing that they need called oxygen, right? So the way that our body gets oxygen is through breathing by inspiring air through our lungs. And it's not like it's getting it pumped up for a show or inspiring it to create new things. No. In this case, inspiration is going to be referring to inhalation of air. And then we will usually use a word like expiration for the expulsion of air. But yeah, all of our cells need energy. And something that we'll talk about once we get into kind of like nutrition and metabolism in a couple of weeks is we'll end up discussing something called the Krebs cycle or glycolysis or the citric acid cycle. And so each of those are components of things like the mitochondria. Sorry, Valerie, I gotta mute you. Oh. Okay. But so the mitochondria are present in all of the cells in our body, and that's going to be what powers most of the energy consumption that our cells need in order to survive. So how do those things get the oxygen that they need? And as it turns out, oxygen isn't necessarily used in the way that you may think it is, but it is an important step in being able to generate ATP, which is what our cells use for a bunch of different things that we've talked about throughout the year. So the way that our cells get that oxygen is through the respiratory system, right? Not only are we able to deliver things like oxygen or other kinds of dissolved gases to our cells via the blood, but we can also rid it of other important gases from cells, things like carbon dioxide. And one of the cool things about our circulatory system is that we're able to bind things like oxygen and carbon dioxide to red blood cells. But as we talked about before, hemoglobin loses or gains affinity for either oxygen or carbon dioxide, depending on how many molecules of that particular gas that it has or that it has bound to it. But what's interesting is most of the carbon dioxide ends up getting dissolved in the bloodstream and then is dispersed into the lungs that way. Okay, so we get oxygen from the air via diffusion across a really delicate surface. It's literally a single cell thick, a single squamous cell thick, so not even like a columnar or pseudocolumnar or anything like that. Single squamous cell thick with a very thin layer of a fluid that we call surfactant, and I'll explain what that is a little bit later. But it's all diffusion. Really, the cool thing that we can do with the second half of this lecture is you can reduce the mechanisms of gas exchange down to concentration gradients, except we give it some fancier names like pressure gradients instead. But really, it's the same concept that we've been talking about throughout the year. Okay, so oxygen diffuses into the lungs via those exchange surfaces that we call alveoli, and then ends up getting carried to the cells via the cardiovascular system, which will also return carbon dioxide back to the lungs. All right, so let's talk about some of the functions of the respiratory system here. So the respiratory system provides extensive gas exchange surface area between air and the circulating blood. So by maximizing the amount of surface area that we have for things like gas exchange, then that also allows us to put volumetric constraints on the organs that are responsible for gas exchange. There's some really cool animals that are vertebrates like us that have spines, but also use other organs in their bodies to breathe through. A good example of that are things like frogs, which can actually breathe through their skin, not just their lungs, which is pretty cool. So the lungs or the respiratory system itself provides a lot of surface area per the volume that it's taking up. So it moves air to and from the gas exchange surfaces of the lungs. Those would be the alveoli, or the little functional units of the lungs. protects respiratory surfaces from outside environment. So the way that we can do that is by adding things like surfactant or a mucus to protect the, well, actually, let me back up. The bronchi, the tubes that deliver or basically go from a large tube in our throat called the trachea, which you can feel when you poke your neck right here. That tube that you're poking is your trachea. That's the main airway that air travels down from your mouth or the oropharynx into the lungs. And so that large tube called the trachea splits off into some other vessels that we'll talk about in a little bit. But along those smaller vessels, we have mucosal layers along with oh my gosh, pseudostratified columnar epithelia that help protect the alveoli from different kinds of pathogens or particles that may be trying to get into the alveoli. So that's pretty cool that it can do that. And so we talked about that a little bit in our last chapter on the lymphatic system. One of the roles of that mucus is to catch different kinds of particles and prevent them from getting into the alveoli, because the alveoli themselves do have a couple little immune system aids that patrol around on the surface level of those cells, but they can become quickly overwhelmed if a cell infiltrates that area effectively. Something like COVID does that very well, and we'll talk a little bit about the immune response that way. But so we have mucosal layers that help protect against most pathogens that can infect or attack the tissue of the alveoli. The surfactant also protects it a little bit, but the surfactant's there mostly to make sure that the alveoli don't collapse on themselves. What I'm doing right now, producing sound, all of that is predicated by me expelling air out of my trachea through my larynx, which is our voice box, and it has some complicated ligaments and muscles that we'll talk about in a little bit. But through controlling how taut or how relaxed those vocal cords are, that's going to determine the pitch of my voice, but all of that is controlled through the respiratory system. And also the olfactory system as well. So our nose is part of our respiratory system and contributes to our sense of smell. All right, so as I mentioned before, we can typically divide the respiratory system into two different parts. We have the upper respiratory system, which is the area above the larynx, and then we have the lower respiratory system, which is everything below the larynx. Pretty much once you get into the trachea and then down into the bronchioles there. So we have the respiratory tract itself, which consists of a conducting portion from the nasal cavity to the terminal bronchioles, and it consists of a respiratory portion. We have the respiratory bronchioles and alveoli, right? So the conducting portion and respiratory portion, two separate components. All right, so here's how we can kind of break that down. So the upper respiratory tract up here, we have our nose and our nasal cavity, our pharynx, right? So our throat, the pharynx can actually be divided up into three different parts that we'll talk about. And then the larynx here, which is kind of where our voice box is. And that's where we've got all these complicated ligaments and things that control the pitch and tone of our voice. And then we've got our trachea that feeds down into the lungs right here. So right after the larynx right here, that's where we get into our lower respiratory tract. And then we have our upper respiratory tract. So different things that can affect the upper respiratory tract infections are things like your common colds, right? Where you have inflammation of various sinus cavities. You can have things like sinusitis. Your maxillary cavities can be inflamed. Your sphenoidal cavities can be inflamed. You can have extra mucus that gets produced as a result of that. So pretty, fairly standard upper respiratory infections, but things like lower respiratory infections, things like SARS-CoV-2, or what it ends up developing into, which would be pneumonia, all would be examples of lower respiratory tract infections. Okay. I have a little blurb here about the nasal conchae. The nasal conchae are pretty interesting. I think I had talked about them a little bit already, but the purpose of the nasal conchae is to humidify air that we inhale through our nose. So those of you guys who are, avid runners and that sort of thing know that you should breathe through your nose as much as you can, especially inhalation, because it introduces less harsh air to your lungs so that the mucus doesn't dry out, the surfactant doesn't dry out, and then you don't have, respiratory issues or lower respiratory tract issues. Sneezing, I have a little blurb here on the sneezing reflex as well. There's some really cool sections in your textbook that you should look into. There's an example of the ACHOO acronym. So it's not just the sound that we make when we sneeze, but it actually is an acronym. So basically our senses of not sense, but our stimulus to sneeze can come from a couple of different things. Some of them is a light response. Others are a pupillary contraction reflex as well that will control our necessity or need to sneeze. Okay, so here we have the breakdown of our upper respiratory tract, right? So we got our nasal cavity and the pharynx, right? So if we just kind of go through this, really briefly. So we have the nasal cavity, we have all of our various conchae up here. With our nares, we call those the external nares, or just the nasal openings there. And then we've got our frontal and sphenoidal sinuses back here. And then we move back into the nasal cavity proper, where we have the superior, middle, and inferior meatus, so the little openings of the conchae right here. So it's kind of hard to tell, but I've got another image here that we'll see in a sec, but these are actually spiralized structures within the nasal cavity here. And then we've got our various tonsils as well. So we've got our pharyngeal tonsil, the opening of the auditory tube, or sometimes called the eustachian tube. So this is what connects our inner ear to the external environment. So this opens up right here within what we would call kind of the back portion of the nasopharynx. So then we have our pharynx as well, right here, this kind of middle portion where we have, again, our nasopharynx just here, the soft palate of the back of the mouth. So we've got the hard palate made out of the maxilla and the palatine bone. And then we have our uvula just here. We've got our palatine and lingual tonsils just here and here, okay? And then we've got the oropharynx just here. So kind of the opening of the mouth as it leads into both the esophagus and the trachea, or the digestive system and the respiratory system too. So they move down into the larynx proper from the pharynx, right? So the pharynx is kind of this like nose, mouth, and like upper throat region, right? So we got our laryngopharynx, which is this bridge between the pharynx and the larynx right here. Then the larynx itself, this is where we have like our voice box stuff going on. So we've got our hyoid bone here, the epiglottis, which is important. And then we've got our vestibular folds, vocal folds. We have our thyroid cartilage, or also called your Adam's apple, and then your cricoid cartilage, and then other kinds of tracheal cartilage that you can see coming down here. All right. And then the esophagus. So your food tube sits behind or posterior relative to the trachea. So it's interesting too, and something that, something to think about as we go through this. The trachea has these rings of cartilage that are kind of embedded within the tissue and give the trachea some support. But the esophagus doesn't. The esophagus is just a squishy tube that runs posteriorly relative to the trachea here. So, something to think about might be, why does the trachea have these extra rings of cartilage that are surrounding it? What could be the reason for that? But the epiglottis here ends up closing as we end up swallowing food or drinking water, those kinds of things. So what might happen if you, swallow water or eat food and it goes down the wrong tube, right? I don't know why I quoted swallowing, because, we all swallow food or we all swallow water when we consume it. Unless you do it in a suppository, but I imagine most people probably aren't doing something like that. But anyway. The epiglottis closes off as a response to mastication and the swallowing reflex. But sometimes what can happen is that flap can actually remain a little bit open, and you can actually have particles of food or water that get into the trachea, and then you have a coughing reflex based around the cilia of those pseudostratified columnar cells that triggers a smooth muscle reflex that creates a rhythmic contraction of the trachea to force those particles out of there and get them back into the esophagus where they belong, pesky food particles. But then I have this little blurb right here. One of you guys sent me this link to a new gland called the tubarial salivary gland. It's not depicted on here. It's not really depicted in any anatomy textbook because it was recently discovered this year. But yeah, it hangs out right about here, kind of right where the oropharynx begins and the nasopharynx ends. So it kind of hangs out right back here, All right. It's a fairly delicate gland from the paper that I read. So on the slides that I posted for you guys, or if you watch this back and you're recording, you can pause the video here, and you can copy out this this link here. But it's pretty interesting. They go into kind of how they found these glands, and it was actually kind of by accident, but they were doing chemotherapy on individuals for kidney cancer, and they ended up whatever radiographic imaging technique they used. It ended up binding to some of the tissue in this gland. So it's kind of interesting. So we have an extra salivary gland that's present there. Kind of cool. Do you have a question, Monteith? I see that you're unmuted here. Nope. Okay. All right, so moving right along. Okay, the nose. Air enters the respiratory system through the nostrils or external nares into the nasal vestibule. The nasal hairs, believe it or not, serve a purpose. They're the first bit of our filtration system that our respiratory system receives, right? So, don't necessarily tweeze or trim out all of those nasal hairs. You need those as part of the filtration system for your respiratory system. So in the nasal cavity, we have that nasal septum that divides the nasal cavities into left and right kind of channels, as it were. We have mucosal secretions from the paranasal sinus and tears that get produced as well. So they can clean and moisten the nasal cavity to kind of wash away any of those particles or foreign pathogens that may get caught in there. The superior portion of the nasal cavity is the olfactory region, which provides our sense of smell, right? So kind of the roof of our nasal cavity, that's where we have those olfactory neurons that are embedded within the epithelia there that contribute to our sense of smell. Air flows from the vestibule into the internal nares, so through the superior, middle, and inferior meatuses. The meatuses are constricted pathways that produce air turbulence. This is where we have the humidification and the warming of air. So we can also trap some foreign particles here as well in some mucosal layers that we produce on these tissues. But one of the main purposes of these meatuses and kind of what they're referred to in other animals that have really pronounced versions of this, or these are the nasal conchae. We have nasal conchae and they're kind of at the forefront of our nose, but in us, the meatuses are the parts that end up serving to humidify and warm the air. So here's what that looks like if we take a frontal section through a human skull. We can see that kind of they have these curved natures of each of the meatuses here, right? So those are the areas where we have this curvature happening. We have specialized epithelia that serve to warm and humidify that air. So just adding moisture to that is nice and dandy for our lungs, and they really appreciate that. All right. So here we've got another sagittal section, mid-sagittal section through the upper respiratory system. We won't go over that right now. So here we have another mid-sagittal section through an elderly individual or just an individual that has dentures. We can see we've got our external nares right here, the hard palate, but this is a good look at the nasal conchae. So we have our inferior, middle, and superior nasal conchae right here, okay? And then the middle, inferior, and superior meatuses are the openings after those conchae right there. All right. So the pharynx. The pharynx is a chamber shared by digestive and respiratory systems, which extends from internal nares to entrances to the larynx and the esophagus. And again, so we can divide the larynx up, or I'm sorry, not the larynx, but the pharynx into three different parts. So the nasopharynx, oropharynx, and laryngopharynx, right? So, and again, the indicators here might help you differentiate are just the prefixes. So naso, oro, and laryngo, right? Determining where you find them. So nasopharynx, oropharynx, laryngopharynx. Nasopharynx at the back of the nose, oropharynx, the back of the mouth, laryngopharynx at the beginning of the larynx. So the nasopharynx, it's the superior portion, contains the pharyngeal tonsils and openings to the left and right auditory or eustachian tubes. The oropharynx, middle portion, communicates with the oral cavity. And the laryngopharynx, which is the inferior part, extends from the hyoid bone to the entrance of the larynx and the esophagus. Some really fun to say words in this chapter. Airflow, right? So airflow moves from the pharynx and enters the larynx, right? So we have a cartilaginous structure that surrounds the glottis, which is our narrow opening. And the cartilages of the larynx are threefold. Okay, so we have three large unpaired cartilages that form the larynx, called the thyroid, cricoid, and the epiglottis. All right. So here we've got our anterior view of our tracheal cartilage right here. So we've got our hyoid bone up here with our thyrohyoid membrane. Okay, superior thyroid notch, and then our major thyroid cartilage. Again, that's our Adam's apple. Cricothyroid cartilage ligament with the cricoid cartilage underneath it, just here, and then rest of the tracheal cartilage down here. Then in the posterior view, we can see a really good view of our epiglottis that's kind of in this position. It's closing off the trachea there. So we've got our thyrohyoid membrane just there as well. But then now we get to see some more of the paired cartilages that are happening or that are occurring inside of the trachea right here. So we have our quadrangular membrane, the cuneiform cartilage, along with the corniculate and an arytenoid cartilage. Sorry, I kind of mess up on that one sometimes. Arytenoid cartilage. So corniculate and arytenoid cartilage, along with the cricoid cartilage, these are going to be the ones that contribute to sound production. So these are the ones that pull on and allow for reverberation of our vocal cords, both the true and false. Okay. So we've got in our sagittal section right here, we can see a little bit of adipose tissue forms around right here, but then we have our thyroid cartilage coming across right here, but then our vestibular folds. So we've got our false one that sits superiorly, and then our true one that sits underneath, right? So those are both bound to the arytenoid and the corniculate cartilage just there. Okay, so the reverberation of those, and depending on how taut or loose they are, is going to determine things like pitch and tone with our voice. So thyroid cartilage, also called Adam's apple, hyaline cartilage, forms the anterior and lateral walls of the larynx. Ligaments attached to the hyoid bone, the epiglottis, and laryngeal cartilages. Cricoid is made out of hyaline cartilage, which forms the posterior portion of the larynx. Ligaments attach first to the tracheal cartilage and then articulate with the arytenoid cartilages. And then the epiglottis, made of elastic cartilage, which is nice and convenient because it can open and close and is pliable. Ligaments attached to the thyroid cartilage and the hyoid bone. Cartilage functions. Thyroid and cricoid cartilages support and protect the glottis and the entrance to the trachea. During swallowing, or during our swallowing reflex, the larynx gets elevated and the epiglottis folds back over the glottis, which helps prevent the entry of food and liquids into our respiratory tract. The larynx also contains three pairs of smaller hyaline cartilages, the arytenoid, corniculate, and cuneiform cartilages. So these ones are paired, whereas the other three are unpaired, meaning that there's a right and left side. So cartilage functions, corniculate and arytenoid cartilages functions in opening and closing of the glottis and production of sound due to their connections to those true and false vocal cords, right? So we've got a good internal view of those. So we can see our corniculate and cuneiform cartilages right here, which again have their connections with true vocal folds right here. And then we have our false vocal folds just here. Right? So we've got a nice view of that inside somebody's throat through a laryngoscope. Kind of neat. So the trachea, also called the windpipe. So now we've moved from our upper respiratory tract down into our lower respiratory tract. So this extends from the cricoid cartilage into the mediastinum. So remember, the mediastinum is that extra cavity by proxy within the thoracic cage, where it branches into the left and right pulmonary bronchi. We have the submucosa, which sits beneath the mucosa of the trachea and contains the mucus glands. All right. So we've got our thyroid gland right here. So this is another cool little cross section through roughly around like C4, maybe C5 of the neck. So we can see we've got our not epiglottis, but the esophagus here, common carotid artery, the inner lumen, or the inner layer of the tracheal vessel here, or a respiratory epithelium, and then the tracheal cartilage separating it right there. Okay, so now we move down into the lungs, right? So we've gone past our larynx, right? Now we're into the trachea, okay? So once we get here, this is fairly straightforward. We just have a large tube that gets progressively smaller as we move out into the lungs, right? So we go from our trachea right here down into our bronchioles or bronchi. We call these, like these first ones, the primary bronchus. And then as those branch out, those become secondary bronchi. And then we have terminal branches that end up, I'm sorry, not terminal, but tertiary branches that are segmental in nature. And then those segments will then branch out into what we call terminal branches that then have our alveoli on them. So moving from top to bottom, they're bigger in diameter at the top. So up here at the trachea, larger in size, but then as we move down into the lungs, they get smaller in diameter, but increase in number. Okay. So here is as good a time as any to talk about the coronavirus, right? Because this is typically where the coronavirus affects most people, right? So COVID-19 ends up generating a viral form of itself that infects other cells and then ends up resulting in a pronounced acute respiratory distress disorder that we call severe acute respiratory distress syndrome coronavirus 2 or SARS-CoV-2, right? So as somebody gets infected with the virus, then it ends up being upgraded to this most times. So what this does is it infects the epithelia of the upper and lower airways with diffuse alveolar damage as the primary pulmonary pathology. So the main effect of people that contract the coronavirus and end up getting upgraded to SARS-CoV-2 is damage to the alveoli, right? You can also find damage in the conducting pathways, so within the bronchioles, within the pneumocytes, alveolar macrophages, and hilar lymph. So all of the immune system support for the lungs ends up getting attacked by this virus. It gets named because of the spikes on the surface of the viral capsule. And the way that it infects us is by inserting RNA into our cells. And then that RNA is interesting. And one of the reasons why the COVID virus is actually a little bit more interesting than kind of like your typical flu virus is most other viruses will actually have to infect or not infect. Well, I guess infect is a good word, but is take over the nucleus. But the coronavirus doesn't actually have to do that. It can just simply take over some of the productivity of some ribosomes that are available in the cytoplasm and then start making its own proteins. And then, Bob's your uncle, and then the virus has spread throughout that cell. What we've also seen is that the coronavirus can also merge or fuse with other localized cells, which we think is probably a reasonable indicator of how the virus will spread across the alveolar epithelia of the lungs. So it utilizes both MHC1 and MHC2 proteins to get into the cells, then utilize ribosomes in order to produce the viral proteins. So as I said, it can cause neighboring cells to fuse together. Typical pathology is, your fever, your cough, your shortness of breath, like your typical, kind of flu symptoms, things like fatigue as well. The alveoli are generally the cells that get attacked. And one of the reasons that this gets so out of hand so quickly is because we have generalized macrophages within our alveoli that kind of patrol and, neutralize various kinds of like normal pathogens that can be found around there. But what this virus does is it's an immunodeficiency virus in a way. It ends up attacking immune system cells, so it limits our ability to fight off the virus effectively. So then what ends up happening is our immune system gets overtaxed, and then we create a kind of overzealous, or we retaliate with a little bit too much force in a way. So our immune system cells, like our cytotoxic T cells, for example, end up killing off way too much tissue, like healthy alveolar tissue, instead of tissue that's only infected by the virus, due to the magnitude of this response, because the rest of the immune cells are kind of damaged and or destroyed as a result of this virus. So, what ends up happening is we end up getting severe scarring on our alveolar tissue as a result of prolonged infection with this particular virus. And there's obviously things that contribute to, this increased susceptibility to the virus. So things like being immunosuppressed or having pre-existing conditions, or any sort of, like respiratory condition like asthma, anything that, affects your ability to fight off viruses and disease is going to, increase the effectiveness of this virus, to say. So, where vaccines come into play, and as of the recording of this, we've gotten some pretty promising stage 3 results from companies like Pfizer that are looking into, mass producing a vaccine. And so, as we all are aware, and we know kind of what vaccines do, but vaccines essentially are going to give us like a low dosage of the form of this virus that isn't able to infect us, but just enough to stimulate an immune response so that our memory B cells can read out the sequence or the genes of the coronavirus, and then be able to produce antibodies that can respond to and eliminate COVID viral capsules that can enter our body, and we can neutralize them effectively before we even express symptoms. So that's the idea behind that. All right. And so obviously I'm not, we don't have enough time to cover everything. You could obviously teach an entire course on the coronavirus at this point. But this is just some stuff on like how it affects the respiratory system. The academic paper that I cited, you guys can look up this author. There's a bunch of different authors on it, but the first author is Martinez et al. It was just published back in September. And then you guys should check out Nucleus Medical Media as well. They've got some pretty interesting and, in my opinion, very well put together infographics on, how the virus affects our tissue and those sorts of things. So definitely check out those sources if you'd like some extra, material. Okay, so moving on to inspiration. This slide is actually a little bit out of place. We'll kind of end up circling back around to this. But when we breathe in air, right, we're actually not really breathing in mostly oxygen. It's mostly nitrogen. Pure oxygen can actually be quite poisonous to us if we end up consuming too much of it. So these values are changing due to things like global warming and climate change. What we end up seeing is we're actually having a little bit more carbon dioxide that's being, released back up into the atmosphere than normal. So the oxygen percentages are actually going down, and the carbon dioxide percentages are actually going up a little bit. I'm not too sure about nitrogen or water, but I know those ratios are changing. And so we're doing things obviously offset that, but there's still, an upward trend in those percentages. So moving right along here, we have our medial view of our lungs. So our right lung has 3 separate lobes. We have a superior, middle, and inferior lobe on the right lung. Then the left lung only has 2. We've got our superior and inferior lobes. Okay, and all the vessels that we're seeing here, we can see our bronchi, but then we can see our pulmonary arteries. Again, the arteries here denoted in blue because they are deoxygenated. And then our pulmonary veins that are denoted in red because they are oxygenated, they are oxygen saturated, right? So on our left lung here, we can see the cardiac impression with the cardiac notch, again, leaving space for the heart to occupy that. We've got the oblique fissure that separates the superior and inferior lobes from one another as well. Okay. And then our hilum, you'll see this word pop up in some other chapters that we'll cover too. The hilum is just the indentation or kind of a little recess where we have vasculature that's either entering or exiting an organ. So the spleen has a hilum, the kidney has a hilum. So you'll see that term pop up quite a bit. So we've got our primary bronchi, right and left primary bronchi separated by an internal ridge, the carina. The right primary bronchus is larger in diameter than the left, descends at a steeper angle as well. So the right primary bronchus a little bit bigger, left primary bronchus a little bit smaller and shallower of an angle, steeper of an angle over here. Okay, presumably because we have lots of our cardiac vasculature that's actually exiting the heart and trying to enter the lungs on this side of our respiratory system development here. So the hilum, where the pulmonary nerves, blood vessels, lymphatics, and all that fun stuff enters and exits the lung, anchored in a meshwork of connective tissue, so all rooted kind of in that mediastinum. The root of the lung It's a complex complex of connective tissues, nerves, and vessels within the hilum. It all gets anchored within the mediastinum. Okay, so then our lungs, right? So we've got left and right lungs are in the left and right pleural cavities. So remember, each lung gets its own little isolated cavity. We have the base, which is the inferior portion that rests on the superior surface of the diaphragm, so the bottom part. And then lobes end up getting separated by deep fissures that isolate them. Remember, the right lobe, or the right lung has 3 lobes, and the left lung has 2. Superior, middle, and inferior fissure, or superior, middle, and inferior lobes separated by horizontal and oblique fissures. The horizontal is going to separate the superior from the middle. Oblique is going to separate the middle from the inferior. Superior and inferior separated by an oblique fissure on the left. Lung shape. Right lung is wider and gets displaced upward because of the position of the liver on the right-hand side of our abdomen. Left lung is longer and gets displaced leftward by the heart forming the cardiac notch. So the left lung actually feeds back down behind our diaphragm. It gets, pretty close to the lumbar region of the spine. So here we've got a coronal section through the chest cavity of an actual human. And so this cadaver has been, has either been preserved for quite some time, or very likely this could be an example of smoker's lungs, which is, unfortunately, I've only, in my cadaver experience is only, I've only dissected maybe like 10 cadavers in my limited experience with that. But a significant percentage of those, and I don't know if this is consistent just across donors or what, but a lot of the lungs that I worked on in the cadavers that I dissected were smokers' lungs. And they ended up looking like this. So we've got our right lung, we've got our superior, our horizontal fissure, middle lobe, and then the inferior lobe down here, separated by the oblique fissure right there. So all of that, that's all middle lobe right there, oblique fissure, then inferior lobe. And then we've got our right and left lobes of the liver down here. This is the pericardium that's covering the heart, right? So in the chest cavity, the heart is pretty well isolated there. Okay. Then we've got our superior and inferior lobes just there. And then we've got our falciform ligament connecting the diaphragm to the liver. Diaphragm is going to be an important muscle we'll talk about in just a sec, but I just thought this was cool to have in here. But we've got this little separated boundary that isolates the pleural cavities from one another too. So we've got our right and left pleural cavities. And here we've got an inferior view of the chest cavity, and we can see kind of the position of the heart relative to the lungs. So here we've got our left lung and our right lung. One of the reasons that we know that it's an inferior view is because of the orientation of the heart right here. So the heart ends up leaning mostly towards the left-hand side of the body, and also the position of the greater vessels that are coming down into the abdomen. So we can see our dorsal aorta and the esophagus just there. Okay, so bronchopulmonary segments, the right lung has 10, the left lung has about 8 or 9. And the walls of the bronchial structure, so the walls of primary, secondary, and tertiary bronchi, as you go from the primary down to the tertiary, they end up containing progressively less cartilage and more smooth muscle. Increased smooth muscle tension affects the airway constriction and resistance, and inflammation of this tissue can result in a condition called bronchitis. Okay. We have autonomic control over the contraction of the smooth muscle of the bronchioles. This helps control airflow and resistance to airflow within the lungs. We also have the ability to dilate these muscles as well. So we can increase the surface, or not increase, but increase the diameter of those passageways, which is caused by sympathetic activation of the autonomic nervous system, helps reduce resistance to airflow that way. And then we can also have bronchoconstriction, right? So this is an example of something like bronchitis could be an example of bronchoconstriction. Bronchitis isn't necessarily constriction of the bronchi, but it's inflammation of the bronchi that kind of has a similar effect to constriction of the bronchi. It's by parasympathetic activation of the autonomic nervous system. And you can also have it be a response to an allergic reaction. So like a histamine release can incur bronchoconstriction or bronchitis. Right. So here is a nice little representation of the bronchiole here with a rich network of capillaries that are running down here, but all of the smooth muscle that's surrounding the cartilaginous tissue that makes up the bronchiole. And then so as we get down into, so right here, we're at like our terminal level of the bronchiole. We can see here we've got all these kind of little golf ball sized shapes that are all stuck on top of one another. It kind of looks like a little bunch of grapes in a way. But these grapes are important because they are our major gas exchange organ with our environment, right? And so what's kind of interesting is you have connections between each of the alveoli as well, but they all serve to exchange gas with our environment. So they're all going to be taking up oxygen and giving off carbon dioxide. But we can see that all of our arterioles, our nerves, our lymphatic system, all of those vessels are going to be running in tandem with one another, right? So we've got nervous input all the way down into kind of the arterial, or not arterial, but the alveolar bed, along with the lymphatic vessels as well. Those are going to be found right alongside the capillary beds here. They've been removed so that you can see it a little bit better, but all of that stuff is present around here, right? And that makes sense because we want to be able to, make sure that we're able to fight off as many pathogens as we can. And also keep in mind that even though we're at the lowest, the most specified level of our respiratory system here, this is still technically exposed to the external environment. All of this air is coming in from the external environment. We've got filtration systems and everything, but that is considered the external environment. So we want to make sure that we have as good of an immune response as possible if we need it, right? Okay, so an alveolus, a single one of those sacs. Respiratory bronchioles, okay. So Leah, I'll get to your question in a little bit here. You've got a lot there, and that's awesome. But let me get through this one. So an alveolus, respiratory bronchioles are connected to alveoli along alveolar ducts. Alveolar ducts end at alveolar sacs, and we have common chambers connected to many individual alveoli, so we can transfer gas in between alveoli if necessary. So extensive network of capillaries surrounded by elastic fibers so that they can retain their original shape. Okay, so here's what that looks like under a microscope, but what we're actually seeing here is the remnants of the areas where the sacs were. What we're actually seeing with this honeycomb structure here is the ligamentous connections that form kind of the skeleton of the alveolar sacs, so not the actual sacs themselves. Okay, so we have a single lobule right here, again, a single little bunch of grapes. And we can see we've got the little sacs that kind of form up right into those ligamentous connections between them. Okay. So here's what our individual alveoli look like. So an individual alveolus or a sphere here is going to be made up of a bunch of simple squamous cells, okay, that are all connected to one another like this, obviously. We've got some important accessory cells too. So we've got some alveolar macrophages that kind of patrol the space in between the cells, so kind of like as a first line defense to any pathogens that get through the mucosal layer of the bronchi or the filtration system of the nasopharynx, all of that stuff. And then we have two different kinds of pneumocytes. So we have pneumocyte type 1 and pneumocyte type 2. I'll explain what those do in a little bit. But this is our general makeup of what an alveolus looks like inside of the alveolar sac. And then we can see all of these little openings here are the capillary bed openings that are going to be where we have gas exchange occurring. Okay. So alveolar epithelia consists of simple squamous epithelial cells, thin, delicate pneumocytes that are type 1, patrolled by alveolar macrophages, also called dust cells. And we have type 2 pneumocytes called septal cells that produce surfactant. And the role of surfactant is to make sure that the alveoli retain their shape as we inhale and exhale so they don't collapse on themselves and then we suffocate to death. So yeah, surfactant, oily secretion, contains phospholipids and proteins, coats the alveolar surfaces and reduces surface tension, giving us all those added benefits. Oops, there we go. Okay, so when we have something like respiratory distress occur, where we have difficulty inspiring or difficulty exchanging gas with our environment, usually this can be due to things like alveolar collapse or inflammation of the alveoli, when our pneumocytes, or type 2 pneumocytes, don't produce enough surfactant and then the alveoli collapse on themselves. And then we have this respiratory membrane, which is our thin membrane of the alveoli where gas exchange ends up taking place. Okay. So here we're moved down into the space where we've got our capillary bed. We've got our air that's passed down here into the alveoli. All right. So now we're going to end up looking at how gas exchange occurs, what controls gas exchange. Okay. So keep in mind these layers of cells and these layers of tissue here, because there's going to be a couple different barriers that are going to contribute to, what we call partial pressures of gases and the pressure of gas in the arteries versus pressure of gas in the atmosphere. So we have three layers of the respiratory membrane. We have the squamous epithelial lining of the alveolus, endothelial cells lining an adjacent capillary, and then we have the fused basal laminae between the alveolar and the endothelial cells of the capillary. Okay, so here's what that looks like. So we've got the airspace inside the alveolus, we have the alveolar epithelium, the fused basal lamina from the cells of either one of these, the surfactant layer, I forgot to mention the surfactant layer inside the airspace of the alveolus here. We have the endothelium of our capillary bed, and then the lumen of the capillary bed, where we can have things like dissolved ions, dissolved gases, all that stuff. Or typically if you're oxygen, when you're bound to a red blood cell. So we've got a few different barriers here that are going to provide resistance to gas exchange. But this is as thin as we can get it. So literally, pretty much, barring these, surfactant and basal lamina barriers, it's literally like single cell separation between the alveolar space and the lumen of the endothelial cell. Okay. So again, this image shows the same exact thing with the inside of our capillary bed, the endothelium of the capillary bed, the basal lamina that are fused right here, the endothelium of the squamous epithelia of the alveolus, the surfactant layer, and then the open air space, right? So quite a few layers right there. And we have to be able to diffuse oxygen, or carbon dioxide rather, into the alveolus and diffuse oxygen into the capillary bed. So how do we control things like that? What's going to allow us to do that? It's a couple different things. And like I said, it all boils down to concentration gradients. Okay. So turns out even with all those barriers, diffusion is quite rapid because the distance is so small. That single cell distance, it works out really well for us. And so we're able to capitalize on that and utilize things like the pressure of gas in our atmosphere, along with the pressure of gas within our vascular system, to be able to force those things down their gradients, as it were. Inflammation of the lobules, called pneumonia, causes fluid to leak into the alveoli and compromises the function of the respiratory membrane. So this is one of the side effects of something like COVID that it can have on our respiratory system. So as the alveolar tissue gets damaged, you can have fluid leakage that hinders the effectiveness of the alveoli. Okay, so let's talk about pressures here. So something like blood pressure, typically in the pulmonary circuit, is quite low. So if you guys remember, it's roughly 30 millimeters of mercury inside the pulmonary circuit. So once we get past that capillary bed, it's pretty low at that point, almost non-existent. Pulmonary vessels, for this reason, can get easily blocked by blood clots, fat, or air bubbles, and can cause things like pulmonary embolisms as they end up in the lungs. All right. So here we've got an superior view of our lungs here. And so just as a reminder, our lymphatic supply follows the nature of the blood vessels, right? So it can be superficial, deep to the visceral pleura, and deep and can follow the actual bronchioles. So we have lymphatic tissue within the pleural space as well as inside of the actual lung tissue itself. Okay, so with breathing mechanics, this is kind of cool. So this figure shows all the different muscles that are involved in breathing, right? So we're all breathing right now, otherwise you're dead, right? Or you're holding your breath because you like to hold your breath, and that's fine. Just don't forget to breathe. But all of these muscles serve to essentially open up or create a bucket handle effect with our ribs, right? So we've got the sternocleidomastoid, our scalene muscles, parts of our pectoralis minor, the intercostal muscles are a big one as well. The rectus abdominis can contribute too. But one of the main muscles for inspiration and expiration is the diaphragm. It's kind of the unsung hero. I wouldn't say it's the unsung hero. It's pretty well understood. But the diaphragm normally, as it's resting inside of both, kind of the abdominal cavity and the pleural cavity, it's the barrier that separates the two of them. As it's relaxed, it forms this deep U-shaped curve that creates a positive pressure on the inside of the chest cavity. And as we inhale and we tell the diaphragm to contract, one of the reasons that we're able to breathe the way that we do is because as the diaphragm contracts, it shortens its length, and so it pulls down on itself. And so kind of the best way to explain it is the volume of the chest cavity increases as the diaphragm contracts. So as the diaphragm shortens, it goes from having this kind of U-shaped curve to kind of being flattened out. And as that happens, it's going to increase the volume of the chest cavity. And so we are able to bring air in or inspire air that way. There's other cool vertebrates that do things a little bit differently. But the way our natural breathing cycle is we are natural exhalers, but we have to think about inhaling, right? Or we don't think about it, it's controlled autonomically, but you can control things like your breathing patterns and your breathing behavior by kind of shifting how your brain controls that. You can take over voluntary control of the diaphragm if necessary. So right now, just there, I was taking a deep breath, a deep inspiration. So my diaphragm was super, ultra, mega contracted. And then my muscles, like my scalenes and my sternocleidomastoids, were all pulling out on the ribs to help increase the volumetric capacity of the chest cavity. So diaphragm, along with these secondary muscles, are going to be utilized for different kinds of breathing behaviors, but it's mostly the diaphragm. Okay. So here I've got some notes on kind of how the pressure system works, so how gas exchange works within our body. So changes in volume result in a change in pressure, okay? So this is something called Boyle's law, which we'll talk about in a second. So as volume goes up, the internal pressure of whatever space or container that you're in goes down. So if you've got a gas that's bouncing around in a container, if you give it more space to bounce around in, there's going to be less pressure there. Whereas if you decrease the volume or you decrease the size of that container, whatever gas that's in there that's bouncing around, the pressure is going to go up because they're going to be bouncing off of each other a lot more consistently. Okay, so as volume goes down, internal pressure goes up. As volume goes up, internal pressure goes down. Air follows its own concentration gradient. So I want to, as a disclaimer, the actual physiological pathology of this is not a concentration gradient in the chemical sense. It's what we call a pressure gradient. But the mechanics and the physics is pretty similar. So it might be helpful to think about it in terms of concentration and kind of ignore the millimeters of mercury and that sort of thing. Just kind of look at the numbers and think of those as like higher and lower, pressures, and think of it as a gradient, so higher or lower gradients. So air flows from high to low pressure, so down its gradient. Greater difference in pressure means a greater rate of airflow. So just think of it as like a steeper curve, right? Or a steeper slope. If you got a steeper slope, your rate of change is going to be faster, right? So at the respiratory cycle, we have the end of expiration where alveolar pressure is equal to atmospheric pressure. So as we exhale out, right? So kind of normal expiratory patterns. As you exhale out, okay, your alveolar pressure is equal to your atmospheric pressure, right? So pressure is net neutral. During inspiration, as we inhale, okay, volume increases, so our chest cavity size increases. So since volume is going up, pressure is going down effectively in the alveoli, okay? So as a result of this, air is going to move into the alveoli, right? At the end of inspiration, the thorax isn't expanding. Alveolar pressure equals the atmospheric pressure at that point. As we expire, okay, volume decreases and alveolar pressure increases and air rushes out of the lungs, okay? And then we go back to the first step there. All right, so pulmonary ventilation is the exact mechanism I just described to you, okay? Atmospheric pressure is just the weight of air and has several important physiologic effects. This is Boyle's law that I was telling you about. All this says is that pressure is inversely proportional to volume. So what that means is as volume increases, pressure goes down. As volume decreases, pressure goes up. Okay, so everything that I just described, this is just that same thing in mathematical form. Okay, so the same idea here. If we've got our container right here, if we make it smaller and we don't change the amount of gas in there, the gas is going to build up in pressure. But as we increase the size of the container, that gas is going to have a lot more room to play around in, so the pressure goes down. Okay, so here, this diagram goes through everything that I just described as well. So end of expiration, alveolar pressure is equal to atmospheric pressure and no movement, right? But as we inspire, volume goes up, so pressure goes down. All right, atmospheric pressure is greater than the alveolar pressure and air moves into the tissue of the lungs. As we, at the end of inspiration, pressure equalizes. As we exhale, pressure goes up. So air goes back into the alveoli and we're able to expel air out of our lungs. Okay. We have something called compliance, which is an indicator of expandability. Low compliance requires greater force. High compliance requires less force, right? So things like how stretchy our lungs are, how compliant they are going to determine how easily we're able to inspire and expire, right? So as we get older, these things change. As we get older, our tissue ends up sclerotizing, and so it becomes a little bit stiffer, right? So it's one of the reasons that explains why it's more, as we get older people sometimes have greater difficulty breathing because their tissue just hardens and deteriorates over time. So their compliance goes down. They're not able to change the volume of their lungs as easily as we do in gas exchange, or exchange of gases, is affected by that. Okay, so the respiratory cycle is the cyclical change in intrapleural pressure to operate the respiratory pump, which aids in venous return to the heart. Okay, so now let's talk about volume of gas within the lungs throughout the respiratory cycle. Okay, so we've got something called tidal volume, which is the amount of air moved in and out of the lungs in a single respiratory cycle. So just through normal breathing, okay, that's our tidal volume. So how much I inspire and how much I expire combined. Okay, I'm going to skip over injury to the chest wall, but you should check those things out. Okay, so let me see. I think I've got a better figure here. I'm speeding through because we're running out of time a little bit. Yeah. Okay, so respiratory volumes and capacity. Tidal volume is the volume of air expired or inspired within each breath or with each breath at rest. Quiet breathing tidal volume is approximately 500 milliliters per person, right? So it's generally fairly consistent amongst most individuals, but again, we as adults are going to have much greater tidal volumes than that of like an infant, right? Inspiratory reserve volume is the amount of air that can be inspired beyond resting tidal volume. And again, this is approximately 3,000 milliliters. So if you just inhale normally, okay, right? But you can inhale roughly 6 times that amount, right? Or 6 times that volume. Expiratory reserve volume, amount of air that can be expired forcefully beyond resting tidal volume. So that's when you exhale maximally, right? And then we have residual volume, which is the volume of air still remaining in respiratory passages and lungs even after maximum expiration. So this is roughly 1,200 milliliters, all right? And then we have some other capacities as well. So functional residual capacity is the expiratory reserve plus residual volume. And then, so we've got a graph here that'll highlight all of these as well. Air remaining in the lungs after normal expiration, so roughly 2,300 milliliters at rest. Inspiratory reserve capacity, tidal volume plus reserve, maximal inspiratorial value after normal expiration, approximately 3,500 mils at rest. Vital capacity is the inspiratory reserve volume plus tidal and expiratory reserve volume. So maximum volume of air capable of being expelled after maximum inspiration. So, right, so full in, full out. And then total lung capacity. Literally all of the air your lungs can hold. All of it, yes, all of it. About 5,800 milliliters, okay? So here's the graph that represents that. So our tidal volume, right, so sitting right about here, so 500 milliliters, okay? So inhale, exhale, inhale, exhale. And then we've got our maximum inspiration, okay? And then maximum expiration, just here. All right. So total lung capacity, 5,800 milliliters. Our vital capacity is roughly 4,600 milliliters. So the maximum amount of air that can be exhaled after maximum inspiration. Okay. And then inspiratory capacity factors in our tidal volume as well as our maximum inspiration. So there's some important clinical things that you can do with forced expiratory vital capacity. Okay, so it's the rate at which lung volume changes during a direct measurement of the vital capacity. Simple and clinically important test where you have a patient inspire maximally and then exhales maximally into a spirometer. The spirometer records the volume of air per second, and this can help identify conditions in which the vital capacity of the lungs is unaffected but the expiratory flow rate is reduced. So this can be an indicator of several conditions like asthma, emphysema, and things like chronic bronchitis. Okay, so what are some things that can affect our ability to exchange gas with our environment? So how thick that respiratory membrane is a good one. The surface area that we're trying to exchange gas with. Generally, the more surface area we have, the easier it's going to be to exchange gas. The smaller the surface area, the more difficult it's going to be. And also the partial pressure of the gases. How much pressure is available to kind of facilitate this concentration gradient, right? So I'm going to skip over this as well, but these are the partial pressures of gases in our atmosphere. So for each of these, so nitrogen, oxygen, carbon dioxide, and water. So in dry air versus humidified, and alveolar and expired air. So you should look at this because what it tells you is that the expired air, the values are actually a little bit interesting. So I'll let you guys take a look at that. And if you figure it out, email me, let me know. Normal partial pressures in the pulmonary vein plasma. So we've got, so pulmonary veins, again, veins that are bringing blood back to the heart that are rich in oxygen and typically poor in carbon dioxide. So 400 millimeters of mercury, 100 millimeters of mercury for oxygen, so 40 of carbon dioxide, 100 of oxygen, and roughly 573 of nitrogen. Okay, so this is the figure that I was telling you about where, you know, it might make a little bit more sense to think about it, and your textbook does a good job of this, is just think about it in terms of numbers, not necessarily millimeters of mercury, right? So up here at part 1, oxygen diffuses into the arterial ends of the pulmonary capillaries, and carbon dioxide diffuses into the alveoli because of the differences in partial pressure, right? So if we look at this, wherever the bigger number is, okay, that's where we're going to have the high part of the concentration. Wherever the lower number is, that's where we're going to have our lower numbers of our lower part of the concentration gradient. So the partial pressure of oxygen in the alveoli as we exhale, okay, is going to be higher than the partial pressure of oxygen within the vascular system. So what happens? As we inhale, we're introducing all of that extra oxygen that was normally in the alveoli into our capillary system, while simultaneously we're able to expel a little bit of carbon dioxide and then get most of that out as we return back into our venous return, rather. So as a result of diffusion at the venous ends of the pulmonary capillaries, the partial pressure of oxygen in the blood is equal to the partial pressure of oxygen in the alveoli. And the PCO2 in the blood is equal to the PCO2 in the alveoli. So as we get out into the blood, okay, the PO2 of blood in the pulmonary veins is less than that in the pulmonary capillaries because of mixing with the deoxygenated blood from the veins, draining the bronchi and the bronchioles, okay? Oxygen diffuses now as we get down to the tissue level out here, so we're at the tissue capillary. Oxygen diffuses out of the arterial ends of tissue capillaries, and CO2 diffuses out of the tissue because of the differences in partial pressure, right? So PCO2 still a little bit lower than it is in the arterial system right here, okay? And then we've got a much higher partial pressure of oxygen in the capillary versus in our tissue right here, okay? Then as a result of diffusion at the venous ends of the tissue capillaries, PO2 in the blood is equal to PO2 in the tissue, and PCO2 in the blood is equal to PCO2 in the tissue. Then we go back up here to step 1, and then we do it all over again. Okay, So now, again, it's the same idea here where you have gas exchange that occurs within the tissues, right? So we have oxygen absorption within the tissue, and then expulsion of CO2 from the tissue into the circulatory system. So in the tissues, CO2 diffuses into the plasma and into red blood cells. Some of it remains within the plasma. In red blood cells, carbon dioxide reacts with water to form carbonic acid in a reaction catalyzed by the enzyme carbonic anhydrase. Carbonic acid dissociates to form bicarbonate and hydrogen. And the chloride shift in antiporter allows for bicarbonate to diffuse out of the red blood cells and chloride ions to diffuse into them, which maintains their electrical neutrality. So again, maintaining that pH balance is important here. Oxygen ends up getting released from hemoglobin as a result of this chemical reaction and diffuses out of the red blood cells down its pressure gradient into the tissue. Okay. Hydrogen ions then combine with hemoglobin, okay, so part 6 over here, which promotes the release of oxygen from hemoglobin. This is what we call the Bohr effect. You'll also see this happen in the reverse, right? So as gas exchange occurs within the lungs. And then we have carbon dioxide that combines with hemoglobin, and hemoglobin that has released oxygen will readily combine with carbon dioxide, and that's what we call the Haldane effect. Okay. So as we get into gas exchange within the lungs, right? So we see our shift in what's being taken up by the red blood cell versus what's being dropped off in the tissue has shifted now because we're in the lungs. And now we want to get rid of all the carbon dioxide that we've picked up from our tissues, and we want to absorb as much of that oxygen as we can. So in the lungs, CO2 diffuses from the red blood cells and the plasma into the alveoli due to that partial pressure gradient. Carbonic anhydrase catalyzes the formation of CO2 and water from bicarbonate here. Bicarbonate ions and hydrogen combine to replace it. And the chloride shift in antiporter allows bicarbonate to diffuse into the red blood cells and chloride ions to diffuse out, maintaining that net electrical neutrality. Oxygen diffuses into the plasma and into the red blood cells. Some of the oxygen remains in the plasma, but most of it ends up actually getting bound to the hemoglobin. Hydrogen ions are released from hemoglobin, which promotes the uptake of oxygen by hemoglobin, the Bohr effect. Carbon dioxide is released from hemoglobin and then is bound to oxygen, or hemoglobin that's bound to oxygen readily releases carbon dioxide, the Haldane effect. So the release of a gas from hemoglobin, Haldane effect, the absorption or the binding of a gas to hemoglobin is the Bohr effect. Okay, so very briefly, we're almost done here actually. So we have several different centers within our brain and in our central nervous system that control things like breathing behavior, okay? So we have respiratory structures within the brainstem that control breathing behavior. We have starting inspiration, where neurons in the medullary respiratory center that are always active are kind of monitoring the start of the inspiratory part of our respiratory cycle. These also monitor blood gas levels. So we have chemoreceptors that are going to determine whether or not we need to increase or decrease our breathing pattern to accommodate for things like increased levels of carbon dioxide within our blood. So increasing inspiration facilitated by costal muscles and the diaphragm. Stopping inspiration is the same neurons that trigger inspiration we can also use to stop it. So we can utilize these things called pontine receptors that we can voluntarily stop our inspiratory mechanism, and then we can go all the way back up and start it all over again. Okay, and so then here we've got several different systems that are going to control things like breathing behavior too. Okay. Oh my gosh, gotta love cats. So the last thing that I'll leave you guys with here is talking about how nervous and chemical mechanisms can affect things like breathing rates. So the green lines represent our increased breathing rates, and then the red lines represent our decreased breathing rates. So all of these are going to come back to the input into the respiratory centers in the medulla oblongata and pons, which will then modify our breathing behavior. So higher centers of the brain, things like speech, emotions, voluntary control of breathing, and action potentials in our motor pathways can both accentuate or decrease breathing behavior. We have medullary chemoreceptors, okay, so within the medulla that respond to a decrease or decline in pH, or which would also mean an increase in carbon dioxide. Okay, so that's going to increase things like breathing behavior. We have the carotid and aortic body chemoreceptors, so decrease in pH, increase in CO2, and also can respond to a decrease in oxygen. So these conditions of hypoxemia and hypercapnia are mutually exclusive. You can have two of them, happen individually or simultaneously. They're not necessarily, if one happens, it doesn't necessarily mean that the other is happening. So each of these chemoreceptors will respond to the presence of those gases, those concentrations, and therefore increase things like breathing behavior. We have the Herring-Brewer reflex, stretch receptors in the lungs, which can modify our breathing rate as well and decrease it. We have proprioceptors in muscles and joints, so responses to things like exercise activity, and also touch receptors for temperature and pain stimuli as well. So if you step on a tack or something, you might notice that your breathing rate goes up in response to that stimulus. Okay,